Hello, Tuskegee University students. This is Dr. Griffin, and today I will be discussing some major themes related to our second week in Biology 315, General Neurobiology. In this second video, I will highlight major concepts in Chapter 4 of your Purves et al. Textbook. This chapter in your textbook focuses on ion channels. And one of the key ideas presented in this textbook is that there is a wealth of diversity of ion channels. Not only are ion channels different in size and shape, but they are relatively selective, some even specific for ion type and size. For instance, there are certain ion channels that only allow for large cations to pass through. There are voltage-gated channels and ligand-gated channels that your book thoroughly discusses, and I will give some insight on in this video. Your book also talks about stretch and heat activated channels. This will not be a major focus in this video. For instance, here is an example of a voltage-gated channel. That means that it opens and closes and changes shape in response to changes in the voltage or potential of the neuron. This particular voltage-gated channel is a sodium voltage-gated channel. You can see here that there are beta subunits that flank the main alpha subunit that has 4 domains. Each domain has some subdomains, 1, 2, 3, 4, 5, and 6. These subdomains give rise to major functions of the channel. For instance, it is theorized, and there is great wealth of support, that the S4 subdomain of the voltage-gated sodium channel is the voltage sensor. The S4 subdomain has many positively charged amino acids, allowing for this S4 subdomain to translocate in response to changes in polarity, meaning changes in potential or voltage. There is the pore selectivity filter. This is only going to allow, given its shape and amino acid composition, sodium ions, to pass through. There are also parts of the sodium channel, particularly the alpha subunit, that allows for modulation, where it can get phosphorylated, that will change shape. Part of the sodium channel that your book doesn't discuss is this site for localization. At this site, N-chiron G can bind, and in binding, it can localize the sodium channel to different parts of the membrane by its interaction with the molecular motor kinesin. Another important part of the sodium channel is its inactivation component, and this allows for sodium channels to inactivate and not fully close, giving a key part of regulation of the classical action potential shape. There are other components too, such as those that bind to local anesthetics, that are depicted in this figure. Now, on top, you see a flattened two-dimensional image of the voltage-gated sodium channel. However, just for pictorial purposes, you must realize that these sodium channels are three-dimensional, and you see how the different domains, 1, 2, 3, and 4, interact with each other, allowing for this pore selectivity filter to come through and be the pore, allow for the pore to be shaped, and allow for sodium, specifically sodium, to flow through this voltage-gated channel. Without this active voltage sensor, the voltage-gated properties are severely affected. There are also ligand-gated channels in our nervous system. Specifically, we would call this an ionotropic receptor. Glutamate, which is a major excitatory neurotransmitter in the nervous system, is able to bind to the receptor, and this ionotropic receptor acts as a classical ion channel. Once bound, the channel will open up and will allow calcium to flow into the neuron. And there are other binding sites as well that will modulate this process. But essentially, this ionotropic receptor acts as an ion channel when it's bound with the appropriate ligand or chemical substance. Ion channels are major sources of medical research, as many small mutations can have devastating effects. One such disease or syndrome that is caused by mutations in ion channels is generalized epilepsy with febrile seizures. It is what we would call a channelopathy, a disorder that has arisen due to mutations or defects in the ion channel. What is most common, and that many of you have probably heard of, are febrile seizures, seizures that occur when a fever happens, particularly in pediatric patients. However, individuals with generalized epilepsy with febrile seizures continue to have epileptic episodes months and years after a febrile seizure. Generalized epilepsy with febrile seizures is a syndrome that is autosomal dominant, which has about a 60% penetrance from generation to generation. And there are at least 2 mutations that have been shown to cause and have been correlated with generalized epilepsy with febrile seizures. And both of these mutations cause a severe delay in the inactivation of voltage-gated sodium channels. Think about this for a minute of how delaying the inactivation of a voltage-gated sodium channel could bring forth seizures or generalized epilepsy. We can discuss more online or in class about how this could occur. Again, just like the first video for week 2 in this course, the goal for week 2 is singular, with multiple subparts, as you can imagine. The major goal for this video and chapter 4 in your neuroscience textbook by Purves et al. Is to relate the structure and function of ion channels to their contribution to neuronal function and/or dysfunction. Make sure that you have watched both videos 1 and 2 for week 2 of Biology 315 that relate to chapters 2 and 4 in your neuroscience textbook. I hope you have a great day. Thanks for your attention.